Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, part of the Finding Genius Foundation. I have Manel Esteller. He's the director. He's part of the Josep Carreras Leukemia Research Institute, and we're going to talk about cancer epigenetics. So, Manel, thanks for coming.
Hello, pleasure to be here.
Yeah. Well, tell me about your research.
So, our research deals with epigenetics mainly. Epigenetics is the regulation of DNA, RNA, and proteins that at the end determine the activity of the genome. So this is a little bit beyond genetics, or the chemical marks that regulate our DNA and our genes. In this area, our main interest is cancer and in cancer epigenetics. That it's a field that, although it started a little bit in the early 80s, it was not until in the close to the 2000s that finally took full speed, and now it's very, very relevant in oncology.
So in epigenetics, I've heard of methylation. I've heard of histone modulation. So what are some of the epigenetic marks that occur, and what does it do? Like, what does methylation do? What does a histone modification do?
Yeah. So if we think in our body, so our brain and our skin, all of them, they have the same DNA sequence, but they're doing different things, and they have different features, the corresponding cells. And this is not associated to the DNA sequence because, as I said, it's the same. The chemical marks in DNA, RNA, and proteins is what distinguish and provide different activities to these different cell types and organs. There are many chemical marks that we call epigenetics. To be called epigenetics, they have to be inherited at least in mitosis, and some of them in meiosis. The main one, or the most popular one, is DNA methylation. That instead of a C in our DNA, we have a methylated C, methylated cytosine. Usually this is associated with the loss of activity of the underlying DNA sequence. And a second mark are histone modifications. Here we have many, but again, probably the most popular is histone acetylation, the addition of a acetyl group to these proteins that we call histone, that they're able to pack DNA and control the activity. So summarizing, DNA methylation and histone modifications are probably the most popular and well-studied epigenetic marks.
Which of them will downregulate a gene more? Methylation is kind of like a localized, direct downregulation, I guess, that prevents transcription. But histone modification, does that affect a wider range of DNA, a wider area?
So what happens here is that many times all these epigenetic marks, they collaborate together to silence genes or DNA sequences. Some of them are tighter, the other weaker, some are faster, and some are slower. For example, DNA methylation is a very stable mark to inactivate a gene. And histone modifications are more dynamic. They change more. If we think about memory, maybe memory relates to more histone modifications, short memories, but imagine that long-standing memories can relate to DNA methylation because it's more stable. In cancer, for example, this is a dynamic process between primary site, metastasis, normal cell transformation. Here, you see changes in both of them, and the most stable are DNA methylation changes. But even those are able to change if we change the microenvironment of the tumor.
So what happens in cancer? What's observed, and at what point do the epigenetic marks start to occur on cancer cells?
If we just focus, for simplification, in DNA methylation, we can see that this is a very early stage molecular lesion. In premalignant tissues, like hyperplasias or adenomas, you can start seeing changes in DNA methylation. Overall, what you see is that cancer cells, they have lost total DNA methylation content. This what creates is chromosomal instability and reactivation of repeats and endoparasitic sequences. But at the same time, this hypomethylation occurs in the context of gaining methylation at very particular sites. And these sites are the promoters of tumor suppressor genes. So in cancer cells, the activity of these anti-oncogenes is not useful because they are not expressed, and many of them are inactivated by DNA methylation. This happens mainly in the promoters that contain what is called CpG islands, that are regions that are very rich in the CpG dinucleotide.
So cancer actually is an increase in methylation of anti-cancer genes.
Yeah, so overall there is a loss in the total methylation content, but there is a particular gain at particular regions. Genes that are anti-angiogenic, they get silenced by DNA methylation. Genes that usually they prevent the entrance in the cell cycle, they get methylated, hypermethylated, and then they cannot prevent that the cells goes to cell division. Genes that are important in keeping the binding between cells, intercellular binding, they are lost by DNA methylation in the promoters, and then the cells are able to spread out and generate metastasis. All these famous hallmarks of human cancer, defined by many authors, all of them, they have in a given tumor 2-3 epigenetic events that leads to transcriptional silencing of genes involved in that particular pathway.
So how do epigenetic changes first arise? What kind of molecules or enzymes are responsible for them, and what initiates it?
Yeah, so in the last, I would say, 10, 15 years, has been produced a very complete characterization of enzymes and different proteins that regulate all this epigenetic setting. So in very easy manner can be divided in writers, readers, and erasers. The writers add the chemical marks, the erasers eliminate these chemical marks, and the readers are proteins that binds to them. There is an equilibrium of this in a normal cell. An equilibrium with changes because you have to adapt. Cells have to adapt to changes in the microenvironment. And in cancer cells, there is some imbalance of all these proteins. This can be DNA methyltransferases, methyl CpG binding domain protein, histone deacetylases, chromatin remodeling factors, and this leads to cancer. Some of this is associated with some sporadic changes and random changes that are selected because they provide an advantage, but also has been found in recent times that some of these genes are also mutated in human cancer. And these mutations that occurs like a mutation in an oncogene, but you have mutations, for example, in a histone methyltransferase, and this also foster the cellular transformation.
Are you able to model some of the first epigenetic marks that appear in various cancers, and are they similar, or is there a lot of heterogeneity in the epigenetic marks as well?
So there is like a common cluster of genes that are frequently methylated across different tumor types. Imagine like a cell cycle inhibitor like P16. So this is inactivated in many tumor types. Imagine an adenine repair enzyme like MGMT. This is inactivated in brain tumors, in lymphomas, in colon tumors, etc. But some are very specific. For example, BRCA1, that is the famous breast cancer family gene, is inactivated by methylation only in breast and ovarian cancer. And another gene that is involved in DNA repair, MLH1, that's involved in the Lynch syndrome, it's only inactivated by DNA methylation in colon, gastric, and uterine cancer. So there are genes that are more general, lost in cancer by epigenetics, and some that are very cancer tumor type specific.
For a given cancer, is there a reliable pattern to the epigenetic marks? Do you again, for a given cancer, do you always tend to see the same epigenetic changes, or is it very variable?
I would say that among the different tumor types, there is a common family of set of genes that undergo changes in the epigenetic setting, but you have each one has a particular change that can define it. If we look, for example, in tumors in kids, in some leukemias and some sarcomas, they have a very particular epigenetic setting that is not observed in adult tumors. For example, there are subtypes of brain tumors in childhood that they have a particular point mutation in a histone that is not observed anywhere else. So it only happens in that setting. And vice versa. For example, a lung tumor in a smoker undergoes DNA methylation changes that usually we don't see in a young person, in a tumor in a young person.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Right, but for a given cancer, and whether that cancer hits, let's say it only hits adults, how reliable are the epigenetic changes? Do they usually happen the same way, or are they very variable?
So the mechanisms are the same everywhere. It doesn't matter so much the age, although it is true that those tumors that appear very early in life, many times there is sometimes there is an underlying developmental defect that can drive them. So imagine a translocation that involves a histone modifier. And this translocation, in part, is driving downstream events that involve changes in the epigenetic landscape.
Well, if you look longitudinally at epigenetic marks of a tumor, do you see a progression?
Yeah.
Or if you take a tumor, and you look at it spatially and you profile the epigenetic marks of the different cells in it, has anyone done that? And what do you see, like longitudinally and spatially?
So this is a very interesting question that we have been addressing in my lab for many years. And we have seen that tumors adapt and tumors change to different conditions, and they change their profile of DNA methylation, for example. When we compare a primary tumor and a metastasis, there is already a 90% imagine of genes that are the same profile of epigenetics, but 10% has changed. And they change to adapt because they have to escape the primary site and grow, for example, in the lymph node. The same can be said when you have a tumor. This tumor receives chemotherapy, and then months later, or years later, the tumor reappear, relapse, and then we look at DNA methylation and we compare one sample versus the other. You can see that many times they are pretty similar, but they have changed a particular set of genes. And this is the set of genes that is critical for the survival of that tumor. These are the genes that create resistance to a particular drug. So yes, there are changes in the tumors that occurs when they are exposed to different external pressure. That can be the generation of metastases, the use of radiation, the use of drugs, etc.
But for a given tumor, have you been able to, again, map out, if let's say a tumor is a sphere, it's 2 centimeters, just making this up.
Yes.
You resect it with surgery. Has anyone tried to look and say, make a 3D model of the tumor and look at the epigenetic marks? What does it look like in the center versus the periphery?
Yes.
And make that map. Have you done that?
So this is a question that now is starting to be addressed with the new tools, for example, using single-cell technologies applied to epigenetics. So we can do this for chromatin, and now we're starting to do it for DNA methylation. But it has been done also using not so single-cell technology. But for example, we have shown in the recent past that looking at in a given tumor, in one patient, that tumor, and selected different parts of the tumor, there are some difference between the parts. 90% of the bulk methylation is the same, but there are 10% different targets. And for example, the most external part of the tumor, the one that's interacting with the normal microenvironment, is the one that shows the most changes. It's pretty interesting that.
Oh, so like the outer shell of, let's say, a spherical tumor is going to be the most changed because that's interacting most with the local microenvironment?
Yes, that's what we saw in the case of colorectal cancer, that this part that is interacting with the normal tissues is the more different part. Remember, this is the part that in theory is growing and has to reach an agreement or is on war with the surrounding tissue. So a lot of chemical communication is going on there.
It would be difficult, but can you get any idea of longitudinally how a tumor will change epigenetically? does the change happen, from the outside in, or is it primarily like an outside shell of change? And I mean, how could you use a model even to look at something longitudinally over, like, let's say, a month?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
A quantification of what's external, what's internal, to the best of my knowledge, has not been carefully done. What we know is that some of these changes are driven itself by the tumor and some are driven by external pressure. For example, as the tumor advances from a premalignal lesion in situ carcinoma, stage 1, stage 4, met in the lymph node, met at a distant region, there is an accumulation of events. When you finally reach the most aggressive tumor that has escaped from the breast cancer, has gone to the brain, this is the most different tumor type. And it's the more different in the sense that has accumulated the most higher number of epigenetic aberrations.
Okay, so it looks more and more different epigenetically than it used to.
That is not so different that happened in genetics. It's very similar progression we're seeing here.
Have you looked at healthy tissue? In it, the healthy localized tissue in addition to a tumor,
yes,
and to see if that's affected as well by marks.
Yes, some of these neighboring tissues and cells has been studied in recent years, and for example, we and others have shown that the fibroblast that surrounds some of lung tumors or colorectal tumors, they undergo some epigenetic changes. Little compared with the ones that undergo the tumor itself, but some are observed there. And it is clear that the fibroblast, very close to a cancer or within the cancer, at the epigenetic setting is different than the completely healthy fibroblast.
What about the localized microbiome around tumors? It would be kind of crazy, but what if, let's say it's composed of, a certain type of bacteria and it, and has anyone looked at the bacteria that, again, surround tumors to see if they have been modified, if their DNA, if their genetic material has been modified in some other way?
Yeah, so it's not so crazy. So let's say that there are some studies ongoing showing the crosstalk between our epigenome and the type of microbiome. This microbiome can be in normal or in cancer. Okay. Different question is if we change the microbiome of epigenome of bacteria. That is very complex. The epigenome of bacteria is something that is really a nightmare for a scientist. Very complicated to study. It's very dynamic. It involves many different chemical mass that we have. But one important and this is relevant right now, is that one epigenome that is very relevant is the epigenome of viral epigenomes. And here we show also a few times that the virus, like hepatitis B or HIV, they are able to change the epigenome of the host cell. But the host cell is also able to change the epigenome of the virus. Again, this is an example of molecular crosstalk between our cells and, in this case, an external agent like a viral particle.
The epigenome, it alters the efficacy, I would think, and the ability of the virus to, let's say, compromise or take over a cell. Like a cell that has different markings would experience a viral infection differently. But what do you mean you say that they are changing the viral epigenome? Does that mean that a cancer cell that's infected by a virus versus a non-cancer cell, they would, I guess, lead to the packaging of daughter virions that are different because of the cell's markings, or how?
So imagine the following. Imagine a lymphoma. Some of these lymphomas are related to EBV, Epstein-Barr virus infection, mainly when you have an immune deficiency. And what happened here is that the virus itself, it has proteins that are able to hijack our epigenetic modifiers. It's able to produce a viral protein that binds to a DNA methyltransferase of the human cell and then brings this DNA methyltransferase to the viral genome and is able to methylate a particular region of the virus. And this can be done, and the reason for this is that in this way, the virus escapes from the immune system. Because DNA methylation is like a camouflage for the virus, something that it helps the virus not to be detected by our immune cells.
But you would see this only, I guess, in the viral progeny that exit a given cell, or do you see that at other points somehow?
So it's when they have to divide, they disappear, and when they get incorporated, for example, those get incorporated in the DNA, they acquire the same type of epigenetic camouflage. Yeah. So in every round of division. It's very interesting. Like, for example, there are studies now in HIV, because in HIV, thanks to the research of many people, so we have been able to, let's say, tolerate the infection, to control the HIV infection, but we're not able to eliminate completely. So there's always a reservoir of virus. This reservoir is there in part because we cannot see it. The immune system cannot see it because you cannot see it because it has a epigenetic modification that makes the antigens undetectable by our system. Okay, and there's some clinical trials using epigenetic drugs to wake up a little bit the virus, and then the retroviral agent can see the virus and kill the virus. And there are several trials now in that area.
Oh, there are drugs that change the epigenetic marks of a given cell type?
Yeah, and this is not something that is in the books or in the labs. And now, for example, in patients, there are approved 9 drugs that are based on epigenetics. These epigenetic drugs, most of them are inhibitors of DNA methylation or inhibitors of histone deacetylases, and there are a couple of inhibitors of histone methyltransferases. They are used mainly in the context of leukemia, lymphoma, and sarcomas. Now, these are tumors that have a very specific type of epigenetic modifications, and they work pretty well in particular subset of these patients. Yeah, and they have been, so this is one of the successes, in fact, of epigenetics is this, that finally, after discoveries by many labs, we have now 9 drugs that work in patients, at least for these tumor types.
Yeah, that's amazing. So have people done a comparative virome study of the localized microbiome with tumor versus, healthy tissue in a given organ?
So this has been done, for example, mostly in tumors that are associated with virus. For example, in liver carcinoma associated with hepatitis B virus, lymphomas with EBV virus, cervical cancer and head and neck cancer with papilloma virus. And then now there are, we started this many years ago in 2009, and following that discovery, now there are hundreds of articles dealing with this subject. It's something that some people are starting to use like a biomarker of progression of the disease, of potential progression of cancers associated with virus.
That's crazy. Once the viruses get epigenetically marked, you said they can hide from the immune system better. Are there any trade-offs that are observed? Can you even tell, how does it change the efficacy of a given virus once it's marked?
There, as you know, there are different stages of the virus, of the life of the virus is very complex. And we know, for example, that some people are able to solve the infection, but some people are chronic carriers of a virus, but they do not experiment the disease, and others they have an acute disorder. A lot of these changes are associated with a different pattern of DNA methylation in some of these viruses. And this is a field that really, it's pretty exciting also.
Yeah, because I would think if you have, during a viral infection, the person would end up with a lot of different variants, I guess, or quasi-species of the virus, because some of the cells that are infected, epigenetically marked differently, and therefore will produce like different viral progeny. So very
some are more diverse. Like as you may know, the the hepatitis B virus, this is originated out of quasi-species, but some like the papillomas are more stable. This is really one or two clones.
Does it tend to affect more RNA or DNA viruses?
This affects mostly DNA viruses, and mostly those that are able to be integrated in the genome of the host. And now that you mentioned this, we're talking about the RNA, I just want to briefly point out that, thinking about the future, other areas that we like to talk a lot about epigenetics, but for the future, a lot of effort is going to be focused in what we call epitranscriptomics, that are the modifications of RNA. So RNA is another beautiful molecule that can be regulated by chemical modifications like DNA. And this is another complete universe, and now it's starting to explode.
What does the epigenetic marks of RNA look like versus DNA?
Some of them are very similar. Some RNA has methylation, okay? But in DNA, it's only in the C, in the cytosine. But in RNA, it happens also in the A's, okay? And in the A's, it happens in 2 flavors, the 1A and the 6A, for example. But also happen in other nucleotides, like in the U's, in the uridine. You can have uridine or pseudouridine, and this changes a lot. This changes, for example, the stability of the RNA, the targeting of this RNA to one molecule of the other. the potential to be translated in the ribosome of that RNA or not. So it's an interesting world that we're also exploring we have been doing this also in the last 5 years.
So inside even normal cells, does mRNA, tRNA, RNAs, does any of that get affected by marks itself?
Yes, and there is, again, like epigenetics, there is a good balance. So the epitranscriptome of neuron is different of an epitranscriptome of a skin cell. It's characteristic and defines one or the other together with the epigenetics, of course. And in disease, there is a disbalance. Some of these RNA gain a mark, some lose the mark. For example, we have identified that in colon tumors, some colon tumors, they lose a chemical mark in a tRNA, in a transfer RNA, and they have problems producing sets of proteins. And sets of proteins, these proteins are anti-oncogenes, they are tumor suppressors. So one reason that some tumors, they lose tumor suppressor genes is because they cannot be produced in the ribosomes because the tRNA is not rightly marked. This happens in tRNA, but happens also in ribosomal RNA.
How do you think, from an epigenetic standpoint, cancer starts, and then how do you think epigenetic marks start? Like, what do you think the whole cycle looks like?
Oh, yes. So this is highly complex. So it can happen just by random chance. The random chance, as you know, increase if you increase the number of times that you bet, okay? And this happens as you age. As you age, in every round of cell cycle division, you can introduce an error, a mistake. Imagine a methylated cytosine in a promoter of a CpG island by mistake. And this creates like a cooperative effect, and then you have more and more, and finally you have dense hypermethylation and silencing. It can also be that you have an external toxic. For example, tobacco, the smoking. Tobacco starts changing epigenetics, and is much later than change genetics, after more prolonged exposition to the drug. And this is very interesting because at the beginning still you can go back because methylation is more dynamic. But you finally have acquired the genetic mutations, the chromosomal breakage, is very difficult to go back to a more normal genome.
But how do you think literally a given mark type happens?
Yeah. So here I would say that selection is one of the critical points here.
Epigenetics though is also a deliberate adaptation. Like if I start to go, I don't know if this happens, but if I start to work out 5 days a week, I bet you that causes epigenetic markings and changes. Or if I start smoking, but does anyone know how that happens?
So let's say there are some things that are more easy to understand. For example, diet. If in your diet, you eat a lot of the universal methyl donor, S-adenosylmethionine, then you have a higher chance to get hypermethylation. Okay? So this is easier. So to imagine. Tobacco, smoking. Smoking can induce by itself, by the compounds, an adduct in DNA. And this adduct, by the DNA repair system, is able to attract some methylation there, because adducts can attract DNA methylation. This can be a way to gain hypermethylation in a tumor suppressor gene. Radiation can create small DNA damage that are able to attract DNA methylation there and to silence it. So there are different, as I said, there are different reasons for this. Imagine a mutation, a transcription factor that it's a transcription factor that is not there anymore, so it cannot bind to a promoter. The promoter is completely naked, and then the DNA methyltransferases see the promoter and methylate the promoter. While in normal conditions, because you have a transcription factor protecting that region, should never get methylated. So there are different examples and possibilities.
Does anyone know if there's certain foods that lead to more methylation versus others, like a methylation diet or epigenetic diet?
So it is said that a lot of the classical, more healthy, probably less tasty foods, like all the lettuce and vegetables, is what they have the higher content of the methyl donors.
As someone ages, in general, do people become more epigenetically marked as they age, or less, or just differently marked?
Yeah. So as we age, we did a study about this, studying nonagenarians and centenarians a few years back. So as we age, we start losing DNA methylation everywhere, and this creates an inappropriate transcription profile. Imagine that a lymphocyte can express a gene from the sperm, something so weird like that, because you have lost the control. And at the same time that you lose overall DNA methylation, you gain methylation in certain sites. A little bit like the cancer, okay? But not so strong like in a completely defined, established tumor. But it's very similar. You have this loss of DNA methylation, and at the same time, you have this particular gain at discrete loci of the genome, mainly targeting tumor suppressor genes.
So theoretically, if you eat certain foods, that could somewhat revert your epigenetic mark profile back to a healthier state.
Yes, this is one possibility. Another possibility is that, of course, in food, sometimes you take too much kilos to get the effect. Sometimes this is not good. But there is very active lines of research using drugs that are epigenetic drugs, and with the aim that they are able to slow down the aging process. These drugs, a lot of them act at the level of histone modification. And in this context, for example, what they are called the sirtuin activators are drugs that are candidates in that area. Sirtuins are one type of histone deacetylases.
How specific can epigenetic drugs be? What are their knock-on effects of unintended methylation or histone modification?
This is one of the key questions. So the drugs that we have right now, they are not very specific. What happens here, for example, in cancer, is that the cancer has a short window of tolerance for these drugs, while normal cells, they have a more wider window, so they can resist better any toxic effect. These epigenetic drugs, they are not given a high level. They are given a low level for a long time. If you receive usually chemotherapy in one day, these are given, imagine, for 2-3 weeks, but a very low dose. And then you're able to restore activity of the genes that were silenced without destroying so much the epigenetic setting of the remaining cells.
Yeah, it would be nice. The higher the specificity they have, the better.
It would be nice, but people have tried for many years. They're able to do this in cells, in mice, in organoids, but to make this jump to the patients is complicated because it will require something similar to gene therapy that, as you know, until very recently in the case of CAR cells in leukemia has not worked. So it's something that in the labs work, but for patients, this specific therapy, epigenetic, is not so good. It's more specific, but it's working in leukemias, subtype leukemia, lymphoma, sarcoma. One important thing here is that we like to do is that instead of doing a more specific therapy, finding a more specific patient, the patient that is the great responder to the drug. These patients, for example, are patients that have a particular set of mutations or particular set of epigenetic changes that when you give the drug, they are highly responsive because they cannot survive with that chemical mark. And we're in this process of this. This is called pharmaco epigenetics, and it's also another booming discipline.
So what are some questions that you're researching that you're trying to answer?
So the questions that we like to answer is to have a complete profile of all the epigenetic changes that are going on in cancer cells and also in other disorders. We have published, for example, in neurodegenerative diseases, for example. The second is to use this knowledge to deliver better biomarkers that are able to predict the outcome of the disease or to select better therapies that can be epigenetic or not, but using the epigenome like a biomarker of response to drugs. And the third one is the development of new drugs. For example, we have recently identified a new inhibitor of a histone deacetylase, number 6, that works very well in a subtype of a B lymphoma. And it's something that we do at the preclinical stage, and in this case, now it's in clinical trials. So these 3 lines of research, I will say, the discovery of the complete changes that happen in the epigenome of cancer cells, the use of biomarkers, and the development of drugs are the 3 main areas that we do in my lab.
Okay. One quick question. Are you able to tell if there's crosstalk between primary tumors and metastases? Because you mentioned there's crosstalk between tumors and the localized microbiome, which is interesting, and viruses. But again, do you see this kind of communication, primary to metastases or vice versa?
Yeah, so we're not exploring so much that. So we're comparing the primary versus the metastasis to see what are the difference. and the metastasis in the lymph node or the metastasis in a distant region like the brain or a bone. The cross-communication that can happen using exosomes or other molecules is something that we're not doing in the lab. But there is, for sure, there can be some type of communication. But even in the same tumor, you can experiment changes. For example, the epithelial to mesenchyma transition, this is called EMT. These are a tumor that it looks epithelial, and in the natural history now became mesenchymal, due to epigenetic changes. And we have studied something also pretty new that is called transdifferentiation. And this is a tumor type that became another tumor type. For example, acute lymphoblastic leukemia will become an acute myeloid leukemia. And this is something that this leukemia does to escape from the use of the drug. It changes completely the epigenetic setting without changing the genetics. The DNA is the same, but change epigenetics to escape the efficacy of the drug. And it becomes like another tumor type. In this case, it was an ALL, and now it's an AML. This happened in other places. A prostate adenocarcinoma can become a prostate neurocarcinoma because it changes the epigenetic setting.
Can you tell if you look at, I don't know if you have any situations where a primary tumor and metastases have been either biopsied or resected, and can you tell that a metastasis came from a primary, or let's say you were able to get like 2 or 3 different metastases. Could you tell that one came before another? Yeah. Does one metastasis lead to another, and what's the order of production of them?
Yeah, you can do that type of, as you mentioned, it can be this type of deconvolution experiment, and has been done, and then you're able to elaborate the natural history of the metastasis using DNA methylation. It has been done with other markers. But your question also may relate to another clinical situation that we have provided at least one answer. And this, in oncology, there's something that's called cancer of unknown primary. And these are patients that they have metastasis, but the medical doctor is unable to find the primary site where it came from. And we have elaborated a tool that using DNA methylation, because DNA methylation is so good to define cell and tumor type, that it's able to say, the sample that you gave me, when I do the epigenetic profiling, it looks like a pancreatic cancer. So the primary tumor is in the pancreas. And this is now applied for these particular clinical cases.
Oh, and then does the doctor go back and look and do imaging and they find it?
Then they go carefully to look, okay, I was not thinking about the pancreas. And they go there in the pancreas, etc. For example, real cases from the hospitals. So a patient that has a metastasis, and when we do the analysis, we say that this met came from a breast cancer, but it was a male. And males, of course, the medical oncologists never think about male breast cancer. And then when we say from the molecular standpoint that this is a breast cancer, they go back to that person, to that male, and are able to find a breast cancer in that male, for example.
Well, that's pretty useful.
Yeah, this is useful. This is one of the first applications that we had with DNA methylation. The other application that is used in hospitals around the world is that for a particular type of brain tumor, in gliomas, the silencing by DNA methylation of a DNA repair enzyme is used to decide the therapy. And this is within all the algorithms that medical doctors use around the world.
Have you looked at tumors after chemotherapy? And what does chemotherapy do to the range of epigenetic marks?
Yes, we have done that. So epigenetics changes the tumor at different levels: cell composition, genetics, transcription, and of course epigenetics. It usually, if the tumor relapses, what it creates is it changes the profile to become resistant to the drug. One example: a tumor that is sensitive, a breast tumor that is sensitive to drugs that they are called PARP inhibitors. They are sensitive because the gene BRCA1 is methylated. If the tumor reappears, now that the gene is not methylated and has changed, and this creates a resistance to the drug. So it's something that it's being done now, and we're recognizing that DNA methylation participate in this creation of resistance upon the use of drugs. Yeah, I wouldn't mention this, but sorry, but an important aspect is that epigenetics also participate in immunotherapy. And we shouldn't forget this, because all these immune checkpoint inhibitors, they act if they see antigens in the tumors. And the regulation of these antigens many times is done by DNA methylation. So now there are clinical trials combining, for example, an epigenetic drug and immune checkpoint inhibitor just to boost the immune response of that given tumor.
Oh, okay. It's so complicated, but very interesting.
Yeah, no, but the immune now it has a very direct relation with epigenetics. But immunity is even more complicated than epigenetics.
Yeah, they all work together.
Yeah.
Well, very good. What do you expect? Do you expect any breakthroughs in the next year or so, or it's just an ongoing process? And who knows?
So some of the breakthroughs can be in three lines. One is if we use these new technologies, then now we're able to implement like single cell technologies. They will provide answers that until now we have done in a very gross manner, that studying bulk samples. Single cells provide a lot of answers in the epigenetic fields. The second is the use of big data. Even in the last years, we have done that, and many other people have done that. I would say this is a growing trend. People are starting to analyze in detail DNA methylation in the context of multi-omics, and altogether provide more sense of why tumors are behaving like that, and how can we attack them. And the other is, as you mentioned, maybe it's more specific drugs that are able only to target this particular modification. Hopefully, this will be linked to cell therapies. Delivering these particular epigenetic modifications is something that hopefully this last one we will not see it this year, but hopefully in the coming years we'll see these more specific epigenetic therapies.
Okay, very good. So Manel, where can people go to find out more about your work?
So they can find in our webpage at the Josep Carreras Institute, at the IJC there, just looking for my name there, and some of our latest discoveries also are in Twitter, and you can find also some of there. I'll recommend lectures, reading some of the reviews that we have done in this area, and I'm pretty easy to find there by internet. And anybody has any question, please, they can ask me straight to my email.
Okay, very good. Manel, thank you for coming on the podcast. I appreciate it.
It has been a pleasure. Thank you very much.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.